Okay, you guys. So we should label these little individual circles as, what's a generic word that we could use to describe those? Not a specific example. A generic term. Jacob? Yeah, so this is a monomer. And we can label each individual ones here are monomers. If we look at, so this is a chain of monomers. And this is a very short, when we start to hook them together, it's a very short what? Polymer. So this is a short polymer made out of two monomers. So that's the generic way to think about it. And then specifically you could apply that. So this could be a short polypeptide, and each one of those circles would be what? What's the monomer of that? It's a short polypeptide. Amino acid. And so that would be a specific example of this. And then they're showing you here what? You're hooking a monomer onto this short polymer. And in that process, what's this oval? This is H2O. And so we are forming a bond between this monomer and that short polymer and elongating that polymer. And so by losing water, what's the name of that process? Dehydration. But all this, the things that I'm saying here, like, the bond being formed and forming a larger polymer, that would be part of the explaining the diagram. I'm just not going to write it all out here. I'm just verbally talking about it here. When the water is lost to make this bond, what actually happens? Remember, an OH comes off of one and an H comes off of the other. That's where the water comes from. That frees up the bonds between the two atoms to form a bond with each other and therefore connect it. So therefore we end up with a longer polymer. And so an example in real life that this happens, for instance, like we talked about carbohydrates in plants. The storage carbohydrate in plant is starch. So starch are chains of, what would be the monomer of starch? The monomer of starch. What are starch's long chains of? Madeline?
Is that glucose?
Glucose, yes, absolutely. And more specifically, it's the alpha glucose molecules. And so when plants make glucose, they make glucose through photosynthesis. So this is a glucose molecule. They make glucose through photosynthesis and then hook them together to form starch molecules to store that glucose molecule. And so this is the process by which they would do that, using a water molecule and so on and so forth. So that would be an example of plants forming starch, let's say. Or us forming glycogen in our liver and muscle cells when we have excess glucose in our bodies. The opposite is going on here. So now we take the longer polymer and are breaking it down into a shorter polymer and releasing the monomer that was once attached. Now we have a monomer not bonded. And so we do that by what process? Hydrolysis. Lysis, to split or to cut, hydro with water. So therefore, that's this little guy here, the oval, is the water. We add water, we break that bond, and when we add water, an OH goes back onto one, the H goes back onto another, and that's how we split and break a molecule down. So, for instance, our digestive enzymes help speed this reaction up. And so our digestive enzymes would break like when we eat starch, so if this was a starch molecule, when we eat a starch, like let's say a bread, and we break that starch down, we add the water and it breaks down into glucose molecules, and then our cells can use glucose for energy, all right? And so you should be able to write all that stuff down and do that off the top of your head. We're gonna talk a little bit later, we're not gonna do this back picture here, but when we talk about proteins, we're going to get a little bit more in depth. We did a little bit of proteins yesterday. but this is a picture that you can do a similar thing with, and to write and briefly describe and so on. And so that can, like I said, it's just an idea for some people to use to help you to study, can be helpful. All right? Okay, so we're going to continue on. The format today is going to be a little bit like yesterday, where we're going to do part notes and then we're going to give you part time to work on this in class. All right, the rest of the calendar. All right, so let's talk a little bit more about antigens here. And I think this is where we left off. And so tomorrow, so today we're going to do part notes. Tomorrow I'm going to collect the pattern matching activity. We're going to finish the notes tomorrow, and Monday I'm going to pass back the pattern matching activity, and we're going to use that to help us to review in our review and kind of go over it and so on. And so Monday we'll be reviewing the testing and Tuesday. Okay? All right? So that's the plan for the next couple of days. All right, so this is where we left off. I think the 4 levels of protein structure. So we had talked yesterday about the structure of amino acids, that there are 20 amino acids, and that we hook those amino acids together to form a chain of amino acids called a polypeptide. And then we talked also about that polypeptides are one way to think of a protein, but really that chain of amino acids folds and coils and stuff on itself and gives a 3-dimensional shape. And that shape determines the protein's function, which can have lots of different functions from, we talked about antibodies versus insulin and so on, all these different functions of proteins. So these 4 levels of protein structure. First one's called primary. And this is that basic chain of amino acids. So it's unique sequence of amino acids. So I'm going to draw next to this here. So what amino acid is hooked to what amino acid? So like glycine hooked to leucine, hooked to arginine, and whatever it may be, a chain of amino acids. And that's the most basic way to think of a protein. And so I'm just going to draw a line. So this line represents the chain of proteins, because I'm going to do some things with that chain of proteins as we go on here. So the secondary structure, so proteins, or the polypeptide, is not just a linear chain. Secondary structures consist of coils and folds in the polypeptide chain. So taking that line, instead of keeping it linear, what happens is it begins to coil, kind of like a telephone cord coil, and it can fold. So this is what the folds look like. All right, so coil and fold. It doesn't really have a real 3-dimensional shape yet. So then what happens in the tertiary structure, tertiary means third. So now what happens to this coils and folds is that they curve on each other. And it says it's determined by interactions among various side chains, which are the R groups. Remember, each individual amino acid, the R groups is what changes between them. So let me draw this. So how the tertiary structure looks is, let's say we have a coil here, and then you might have a curve, and then you might have another coil, and then you might have a pleated sheet like this, and so on. And so it starts to curve upon itself. And why it does that, remember, this line represents a chain of amino acids. So what makes it curve like this? Why doesn't it stay linear like this? And it's because there are amino acids in this chain. Let's say that there's an amino acid right here, and it has an R group hanging off of it. That's an R. And then later on down the chain, here's another amino acid right here, and its R group is hanging off. And notice it says it's determined by interactions amongst the R groups. So what happens, and we'll see later on, that R groups can bond together, be attracted to one another if they have charges, and so on. And so if these R groups that are hanging up are attracted to one another, they're going to come together, and that attraction helps to stabilize that curve. So therefore, this chain has a curve here. And the same thing could happen in this curve, and so on and so forth. So that's the tertiary structure. Now, oftentimes in your book, in the enzyme picture, yesterday was the enzyme was a protein that was a big purple structure that had a specific shape. So oftentimes they'll kind of, if I just show you here, like put a shape like a blob and say this is the shape of the protein, but really the shape is determined by this this curving of the amino acid chain. So this would be kind of the curve here, and so this is a specific shape. And so some proteins are finished here. Some proteins are finished in construction here. So meaning that some proteins are only made out of one polypeptide chain. So this would be the finished product. So let's say this is an enzyme, all right, and so it has a specific shape. That shape is going to determine its function. And so, but not all proteins are finished here. Some proteins, as we talked about yesterday, are more than one polypeptide chain. This is one polypeptide chain. That's for proteins that's true, they exhibit what's called the quaternary structure. Quaternary means 4. So we don't stop here for this. It consists of multiple polypeptide chains. So for instance, so what that would mean is like we'd have, here's one chain, let's do here, here's another chain, here's another chain, and here's another chain. So what happens is to make the full protein, you have one polypeptide chain, which is kind of like a subunit, another, and another. So this one has 1, 2, 3, 4 polypeptide chains that all have to come together, and this is one finished protein. An example that I used yesterday, anybody remember the example that I used yesterday that this is the case for? Yeah, it's hemoglobin. So I'm going to put here an example is hemoglobin in your blood. It has more than one polypeptide chain. And so some proteins, that's the case. So some are finished here, some have the quaternary structure. So those are your 4 levels. And so this is another way to look at this picture of the 4 levels. Here's your primary structure, your chain of amino acids. This is showing you the secondary structure, the coils and the pleats, and we'll talk about each of these more specifically here. Here's the bending and so on, and it's usually in that tertiary structure that is really we get the 3-dimensional shape coming on. And then some proteins have that quaternary structure, the more than one polypeptide chain. So we're going to look at each one of these specifically here. So the primary structure is the chain of amino acids, the sequence of amino acids. So I used this analogy yesterday with letters in a word. Same idea, there's 20 amino acids. It depends upon what amino acid is hooked to what amino acid and so on to determine how it's going to coil and how it's going to form that 3-dimensional shape, and that's what determines its function. And so, so the chain of amino acids is really important to get that right shape. Now, how do we know how does your cells know what amino acids to hook together? This is determined by your inherited genetic information, otherwise known as, what's the genetic information? DNA. So this is our DNA here. So we get half of our DNA from mom, half of our DNA from dad, and that DNA contains instructions and tells your cells what amino acids you're going to hook together. So when we make that hemoglobin molecule, we have to make 4 different polypeptides. So how do we know what chains of amino acids? How do we know to hook leucine and glycine and then arginine and so on? And what order of the amino acids? It's determined by your DNA. All right, so the DNA codes for that. And so here we have your chain of amino acids. Notice here that there's the amino end and the carboxyl end. I want to talk a minute about hooking these guys together and why there's an amino end and carboxyl end. So let's do, I'm just gonna put amino acid number 1, a generic amino acid, so it has that central carbon atom. And remember, on one side it has the amino group, on the other side it has the carboxyl group. Off of the third bond it has the H, and off the fourth bond that's the part that changes between amino acids, and that's your R group. Now if we want to hook another amino acid to this to make an amino acid chain, so I'm going to write here amino acid number 2. So I'm going to just do another generic one here, carbon in the middle, NH2 here, C with the double bond O and the OH, H in the R group. So here's my 2 amino acids. When we build an amino acid chain, we want to hook those amino acids together. How do you suppose we do that?
Is it possible that it flips and then the 2, I don't know, not, I would say like the OH and H bond and then the O.
Okay, so you're applying it from like what we did with carbohydrates, right? Yeah. Yeah, you're right, that we lose an OH and an H. For amino acids though, it's a little, where the OH and the H come off, the OH actually comes off of here and the H comes off of here. So it's a little bit different for these guys. And so it loses the water, so we're on the right track. That's called dehydration, right? The dehydration reaction. So now, how are these 2 amino acids gonna bond? Between what 2 atoms? Kobe?
Nitrogen, carbon.
The nitrogen and carbon. So now this carbon is going to bind to this nitrogen. And this was called a peptide bond. All right, the bond between them. And so, so those are 2 amino acids hooked together. And so that's what's going on here. So when we see a chain of amino acids like this, see the end, one end is going to have the amino group hanging off, the other end will have the carboxyl group hanging off. All right, and so we can keep on adding amino acids. All right, so that's your primary. The secondary, the coils and folds of the secondary structure result from hydrogen bonds between repeating constituents, that means chemical groups of the polypeptide backbone. So secondary has the hydrogen bonds is really important here. And we have specific names. So the coils, like the telephone cord here, is called the alpha helix, so it has that helical structure. And then the folds are called beta pleated sheets because it looks like they're pleated, all right? So your picture showing you this. So here are your alpha helix. Now, what this picture is showing you, the blue is like the ribbon, like that's my like blue line that I was drawing here, but it's also showing you the atoms. And so let me just point out here, so this is where, this is a chain of amino acids. So here, this is where, this is one amino acid, the end of one amino acid, hooked to another amino acid, and this is hooked to another. So the lines are the where the amino acids are hooked together here. So what I just wanted to point out here, looking at this, here's one amino acid, here's that central carbon atom, here's the carboxyl group, but remember it lost that OH to bond with the amino group with the next amino acid. And then here's your hydrogen in your R group, and here's the amino group across from the carboxyl group. So that's how we look at this, and this is a chain of amino acids. And so notice here also what's stabilizing and making this coil is, notice that the amino group and the carboxyl group here, from this is one amino acid and this is another amino acid down the line here, but they hydrogen bond, that's the dot 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 here. So they hydrogen bond to one another, so all these dot dot dots are hydrogen bonding, and that stabilizes, and that's what causes the alpha helix. On the flip side, these are your beta pleated sheets. They look like pleats. Let me kind of just illustrate that because it's kind of two separate things to show that it's really, can really be part of one long chain. Let's say my line is the chain of amino acids. So let's say it comes over here, and now these are amino acids, and then it comes over here, and this is the chain of amino acids, and so on. And so I want to show you here how these beta-pleated sheets, how they're hydrogen bonding to one another. They curve and become parallel to one another, and then they hydrogen bond to one another. All right, like that. So those are your hydrogen bonds, and that's what stabilizes those pleats. So the helix and the pleats are the secondary part of this. And then tertiary is the third. It's determined by interactions between R groups rather than interactions between the backbone. So these guys are not hydrogen bonds necessarily, but the R groups interact to one another. And so we're going to look at these different ways that the R groups can interact with one another. But I'm going to wait and discuss all them until tomorrow. So one of those interactions is called a disulfide bridge that we'll look at in the next picture here. Reinforces the protein's conformation. What does conformation mean again? It's shape. So let's better write that here. All right. A lot of blank looks there. So shape. Disulfide bridges. All right. Remember that these are interactions between R groups. Disulfide bridges. Di meaning two. Sulfide should contain what atom? Sulfur, right? And what functional group contains sulfur? The sulfhydryl. And so that looks like an S and an H. So we're going to see that the disulfide bridges, the R groups have that sulfhydryl group. All right, and so we'll look at, that's what this picture here is showing you. It's going to illustrate the interactions between the R groups, but I'm going to hold you in suspense till tomorrow with this, and we'll pick this up tomorrow because I told you I'd give you some time in class to work on that pattern matching. And so I will do that. So I'll let you do that. All right?